Okay, so today we are going to discuss the methods for molecular epidemiology and population genetic studies in Toxoplasma. And in principle, all those methods apply to other, say, pathogens or organisms infect any animal or human hosts. So for molecular studies, of pathogens, there are different ways or different levels of detection or identification. So the first level is, say, let's say it's called a detection. So you want to detect Toxoplasma infections. And so there are two different ways. And one is from the host perspective, and the other one is from the pathogen perspective. So eventually, the goal is to make a diagnosis. So is this animal or say human infected with a particular pathogen or not? So at the end, you get the result, say positive or negative. Okay, so if you look at the host side to identify or diagnose infection, often you use serology test. There are many different tests. So MAT test, the modified agglutination test, we have been using it for a long time. And that's a very simple way to do that. And then there's an indirect fluorescent assay to test the antibodies. And also there's an ELISA, and there are other, say, different flavors of method to detect antibodies. So when there's an infection, there's an immune response to produce antibodies. And if you see antibodies in the host, that imply the host, say, either being infected before or now carry the chronic infection. So that's on the host side. And you can also detect the pathogen. So for us, for example, you want to detect, say, Toxoplasma, and you can isolate the parasites. It's a more complicated way. Or you can just say, let's use molecular method to detect Toxoplasma gondii specific specific, say, DNA, so or genes there. So this is the commonly used, say, as the PCR method. So PCR detection of some sequences. And in order to detect, say, Toxoplasma, and you want to have a higher sensitive PCR test. And for this reason, often you target at repetitive sequences. For example, in Toxoplasma, there's so-called 529 BP element. It has about 300 copies. And the other ones, like the ITS or 18S ribosomal RNA gene, it has somewhere around 100 copies, or the B1 gene, which has about 35 or so copies there. So in order to have a higher sensitivity, you want to use those repetitive sequence because it's easier to detect and more sensitive. So that's for detection or, say, diagnosis. And the second method or way is to identify a particular strains. So the diagnosis give you yes or no a result. But if you want to know more about who is infecting the host, then you want to have more identity or identify the strains. And then you need to go deeper. And this is so-called genotype, genotyping there. And for genotyping, basically you look at DNA. And there are kind of different types of DNA sequences you look at and to get information. And for genotyping, you work on single copy sequences. So instead of, say, for diagnosis, you want repetitive sequence, or you want to sequence with multiple copies. So that's easier to detect. But for identification, you want single copy sequences. So that'll give you kind of clear result. You don't have to deal with the ambiguity of the repetitive sequence there. So the first, the commonly used markers are so-called PCR-RFLP markers. And then there are, say, multilocus sequencing typing, MLST. And the third one is multilocus microsatellite markers. And the fourth one is the whole genome sequencing. So those methods goes with the start with the lower resolution to high resolution, and that is based because of the nature of those sequences. So we can see it from the next slide. So let's say the 4 different methods. And start with the top is the multilocus PCR-RFLP method, and at the bottom is the whole genome sequencing. So you start from the low resolution to high resolution method. And so which method to use depends on what kind of questions you are asking. So for many applications, the low resolution is good enough. And for some application, you have to know very high, well, you have to know lots of sequence information to get a high resolution. So let's say here. So the multilocus PCR-RFLP is based on single nucleotide polymorphism. It's the SNPs. So here we have let's say three samples, A, B, C, there are 3 samples. And if you look at a fragment of the sequence, so here is a stretch of sequence, you line up all 3, you'll see for majority of those sites, the nucleotides are identical. So those are the identicals, but you do see a few red lines or columns here to show different nucleotides. And those nucleotides, a given position or site, is a single nucleotide polymorphism. Polymorphism because they are different. Okay, so how you use those information? So if you use PCR to amplify sequence, say ABC from ABC, 3 different, say, parasite strain, and then if you use restriction enzyme, well, here we have 3 differences there. But if you use restriction enzyme, there are only, say, 1 restriction enzyme available to recognize here the AGCC. So AGCC for the 2 samples and the GGCC for the third one. And the one restriction enzyme can recognize AGCC, then it can cut it. So cut both of the sequences. And then for A and B, it becomes 2 fragments. And then you run the gel electrophoresis, you will see the different bending pattern that tells you the difference. So, however, there are only limited number of restriction enzymes can recognize limited number of, say, variations there. So for this particular example, the other 2 sites, there's no, say, existing enzymes can cut them. So then you cannot tell the differences. So, however, if you look at the whole, if you sequence all those, say, all 3 strains, you get all the sequences there, and then you can use the sequence data and to compare the differences, and then it is the, say, sequencing typing. If you use multiple, say, loci, and then it becomes multi-locus sequencing typing. So that increases the resolution comparing to the PCR-RFLP based method. Okay, so what is in common between the, say, the multilocus PCR-RFLP and the multilocus sequencing typing? The common kind of point is they all look at the single nucleotide polymorphism. So in that sense, they are kind of using a similar data set. And so if you look at the mutation rate of those single nucleotide polymorphism, and the mutation rate is very low. So 10 to the 9th per nucleotide per generation. That means it takes a lot of replication to generate 1 difference there. So that's why, in general, the SNP-based say typing has a lower resolution there. And the third type is the multilocus microsatellite, genotyping. And the microsatellites are short nucleotide repeats, normally 2 to 6 short nucleotide repeats. So here is an example. You have 3 samples, again, A, B, C, and in the middle, you'll see they are, so in red letters, you see the AT repeats. And for each sample, they have different number of repeats. So when you design primer on the flanking region, the conserved sequence there, you design primers, and then you amplify this fragment, and then different samples will give you different sizes of the fragment. So that can be used as a marker to differentiate different strains there. And the last one is the whole genome sequencing. So you sequence the whole genome, and then you can see a lot of differences there, and then it's easy to identify individual strains there. So that's the overall of those molecular method. So here is a quick example to show the multilocus PCR-RFLP genotyping. So here there's a collection of 10 markers, and sometimes we say 11 because for the SEG2 we have 2 fragments. There's alternative SEG2, there's another SEG2. If you count them as one locus, and then we have 11, loci here in this panel. And you will see there are 3 different samples, 1, 2, 3, and the PCR products being digested, and now generate different binding pattern, and then you can tell the differences. So each pattern is called one allele. So here listed at least 3 alleles for those markers, and you combine all the multi-locus markers together to have a higher resolution. So even though the PCR-RFLP in general has low resolution, but if you use multi-locus, you include many different sequence fragments, and then you can still get a decent resolution to identify different isolates there. So the advantage of this method is it's simple, it's fast, and as long as you have PCR machine and the associated equipment, you can do it. So it is not limited by equipment. And also you can do a large number of samples simultaneously. So we often use 96 well plate to run samples. And so in general, it's simple and fast. Okay, so let's come back to the microsatellite. So we just mentioned the microsatellite, a short nucleotide repeat. And here is a different illustration here. So you have sample A, B, C here, and in the middle you have short repeats. And because of the different number of repeats among different samples, and then if you use PCR to amplify this this region, you'll see different size of that PCR product. So that's this is a one microsatellite. So sequence here. Okay, so then a little bit of basics of the biology, how microsatellite being generated. So well, we did not, we kind of forgot to mention, for microsatellite mutation, the mutation rate is very high. So for every 100 to 10,000 replication, it can generate a mutation. And those mutations are called insertion or deletion mutation, and then generate the size difference in terms of the repeats. So how those repeats or insertion or deletion of those repeats being generated. So here is the illustration. So at the top, you have a normal, you have a sequence here, and for there are 1, 2, 3, 4, 5, 6 GTC repeats there. And so then the bottom one, the green, blue one is the template. And the top is the newly synthesized sequence. So if everything goes well, the DNA polymerase will incorporate all those nucleotides and to generate new strand based on the template. So for here, you have 1, 2, 3, 4, 5, 6 copies of CAG. That's normal replication. Okay. However, if there is say a slippage, so during replication, if the alignment of newly synthesized sequence slip along the template string, and then it can generate insertion or deletion. So here is, well, on the top is the newly synthesized, sequence there. If this sequence backward slip, so you see the CAG, instead of at its correct position, it slip backward. And now it forms a here, so bubble here, and then realign to the repeats, and then complete the replication, and it will generate a new sequence there. So this sequence has 6 + 1 repeats. So add additional repeats there. And if that one is used as template for further replication, and now you will have say new sets of say 7 repeats there. So that is insertion. But the original one, the blue, the original one sequence still say can be used as the template and synthesize the original say 6 repeats there. So that's the normal case. And there's another kind of a way of replicate here. So it is called forward repeats. So again, the bottom is the template, the top is newly synthesized sequence, and here there's a forward slippage. So the newly synthesized, say CAG here, it move kind of forward to the next, say GTC for pairing, and then skip one sets over there. And now the newly synthesized sequence will miss 1 repeat and to have 5 repeats instead. And that one, the newly synthesized, can be used as a template to synthesize the complementary sequence. And now you have 5 repeats, so you lose 1. So the frequency of those slippage replication is high. So that's why the microsatellite give you much higher resolution in identify individual microbes. In this case, it's the Toxoplasma. Okay, so now we get back to the using, say, microsatellite to identify Toxoplasma gondii. And here is the paper describing the methodology. So in this paper by Dr. Eisenberg and Dr. Dade in a group, and they developed this multilocus method using 15 microsatellite markers and in a single PCR-assay. So instead of run 1 microsatellite at a time, here this method run 15 altogether in 1 single PCR. Okay, so then how you detect the data, or say collect the data? So first you use multilocus PCR of 15 microsatellite markers, and then you put that PCR products into a sequencer, say the automated sequencer there, and then you detect this fragment, the PCR product, the fragments. Okay, so in this method, there are 15 markers, 15 microsatellite markers, and those markers being divided into 2 sets. And the one set is called typing markers. So they are all, say, dinucleotide polymorphism, so 2 nucleotide repeats. And so for this set, overall, they have less variation. So they have less variation, and they are called typing markers. And if you look at those, say, primers, you'll see the forward primers of each set is labeled by fluorophore. So the first one, table 2, the marker, it is labeled by FAM. And so there are quite a few of them labeled by the FAM, and the others labeled by hex. And total, there are 3 different fluorophore used to label those markers. So here is the so-called typing markers, and in general, they have lower polymorphism. And the second set of the 15 markers is called are called fingerprinting markers. And those markers in general, they have a higher polymorphism. They have a lot of differences there. So they give you more resolution in identify the different Toxoplasma strains. So they are called, say, fingerprinting markers. Again, you see they are labeled with FAM, hex, and NED here. So all the, say, PCR products will be eventually labeled with fluorophore. Yeah, okay. Okay, so if you say just run the PCR product, so you have a multiplex, microsatellite, PCR, you run through the machine, you get it, then you run the product on, say, regular agarose gel. This is what you see. So here we have 1, 2, 3, 4, 5, 6, 7, 8 different Toxoplasma strains. And you use those DNA samples and run the multiplex of the 15 different microsatellites in it. And if you run the product here, this is what you see. So, and for a given, say, strain, it should have 15 different products there with different sizes there. So for this kind of results, you can just say they are there, and you cannot get any information out of it because the agarose gel cannot tell you much difference. So if you look at the very bottom band, so there are 8 different, say, Toxoplasma strains, and you see the bottom band, they have different sizes there. So from low to high here. Well, there's a high one probably here. So the range of this particular marker is 79 to 123 base pair. So that's the difference there. So even though you can see the difference there, it's very hard to call them to see the differences between 2, because sometimes you just cannot have enough, say, resolution to tell the differences there. This is marker N61. So then how you collect the information and see the differences? And well, this is another gel picture to show a lot of samples. You run the multiplex microsatellite markers, and you see a lot of products there, but it's hard to collect any information from it. So that is why those primers are labeled with fluorophore. Using fluorophore, you label your PCR products, and then you run those products in, say, the capillary gel electrophoresis using the, say, the sequencer. And from there, you can see the differences. So the fluorophore, in general, they can say, here is to show the fluorophore with different wavelengths. So for example, a given fluorophore, it can absorb the light at the wave around, let's say, 450, 60-ish here. And then it reflects, they give off light, which is fluorescent light, and then you can see it. You can see it. So it's like, for example, you drive on the road at night, and it's hard to see things on the side, but often you see the traffic, say there's some signs there, and they are bright. The reason they are bright is because your headlight from your car, shed the light onto the fluorescent chemical on the sign, and then it emits back different wavelengths, and then you can see them. So basically, this is the kind of the same thing. Yeah, just to remind you, the primers of those microsatellite markers are labeled with fluorophore, and then you use them to detect the samples. So here's a illustration of multiplex, say microsatellite PCR product run through the capillary gel electrophoresis through a machine, sequencing machine, and then you detect the signal. So you load your sample to the capillary here, and then you apply the electricity and to run it through, and the smaller band will run fast to the bottom quickly, and then the higher bands, high molecular weight bands will run slower. And when they pass through the window of detection, here's the laser light, strikes through the capillary tube and can detect that fluorescent light. And that signal is recorded or detected and recorded by the machine, and then you can see the peaks over there. So that's how you basically do microsatellite typing. You have to use the sequencing machine to collect the data. And okay, so here is an example from that 2010 paper by Dr. Eisenberg and all. And you'll see these figures show 15 different markers there. You see the blue peaks, and there are, I think there are 7 blue peaks there labeled with FAM, fluorophore FAM. And there are 5 green peaks there and 3 black peaks there here. Okay, so all 15, peaks being detected, and then those peaks will be, called to determine their size. So remember, microsatellite is based on the size difference of the product. So how you call those differences there? And you add internal standard there. So the red peaks here, 1, 2, maybe 3 there, 3, 4, 5, 6, 7, 8, 9, 10. So they're internal, fluorophore markers to represent different sizes. So those standards are used to generate a standard curve, and all the signals from those samples are used to compare to the standard and then to call the sizes of different peaks. Let's see. Okay, well, here's that.
I have a question.
Okay, go ahead.
What are the axes on this graph?
The axis is the intensity of the fluorescent light.
Okay, so, yeah. Okay.
Yeah, so the more PCR products there, the higher the peaks. Yeah, so it's the amount of PCR products. Yeah.
So like this N61 has
a lot.
At 50 base pairs.
This one probably is around maybe 90 or 80 range.
Okay.
Yeah. This is
so then each peak is like, so this one has 90 repeats.
The total length of that fragment.
Okay.
Not necessarily being the number of repeats, because when you design primers, the primer is on the side, and between the primer and the repeats, there are some sequences there.
Mm-hmm.
So this size is the repeats plus the normal sequence there.
Okay.
Yeah. Yep. So yeah. So from here you can see N61. There are lots of products. Yeah, kind of a shooting to the roof from this graph, and so yeah, you have the internal control to generate a standard curve, and then from the samples, the samples compare with the standard curve, and then you call the sizes of the different peaks. That's essentially the method here. So there's another paper from the same group, and they use, for this one, they published a paper earlier, so that's in 2002. They used the same idea. So they used 8 microsatellite markers and 83 samples from human patients, and they look at their, relationship. And from here, you'll see they can divide the 83 samples into 3 major groups, and group 1, group 2, and group 3 here. So, and if you look at the phylogenetic tree here, the group 1 is different, is highly different from group 2 and 3. So group 2 and 3 are closer than they are to the group 1. So which is also kind of illustrated on the left side of the graph here. So from this, say, phylogenetic tree, you can see they can almost identify to the level of individual isolates. So there are very few of them share the same node. So most of them at the individual level. So this is to show the microsatellites, they are very good in terms of identify individual strains, because they have a higher resolution. Remember, we talked about the resolution, and the microsatellites, they give higher resolution. So that's why when you want to identify individuals, you very often you want to use the microsatellite instead of using the RFLP. So the point is, say, show here. So in this study, 106 samples were collected from human and some animals there, and there are many of those samples are overlap with the previous study there. And if you look at the phylogenetic tree here, in this case, use PCR-RFLP, but it's only, well, it's 6 markers. So 6 are PCR-RFLP markers, and it can, say, divide those samples into 3 groups, just like the microsatellite. So type 1 group, type 2, and type 3. And also from this phylogenetic, tree, you can see type 1 is more different from type 2 and 3. So 2 and 3 are closer. So if we go back to see the microsatellite data, you'll see the PCR-RFLP can capture the overall, say, relationship, but it has less resolution in identify the individual ones. So that's the, in terms of methodology, that's the difference. So then you may ask, why don't you just use microsatellites for all your studies instead of using PCR-RFLP? And well, when we talk about the process, how you collect the data, you'll see you have to do, say, sequencing. In order to get the microsatellite, you have to do sequencing, and then it may cost more, or you have to have the sequencer to do the study. And so that's the kind of the differences there. And if you want to have the ultimate, say, resolution, then you go with the whole genome sequencing, and which is, well, still expensive, particularly for the genome of Toxoplasma, which is 65 megabytes. Well, it can be quite expensive if you want to do the whole genome sequencing. So that's basically the methods, kind of in a quick review of the different methods, and then it's about it. So the different methods have different use, and you can choose based on the need of your work. So let me stop